my name is Lucia Martinez, and I will be introducing Table 5 Genetics Lab presentation called A Scientific Study on Coral Bleaching and its Relation to Exaptasia.
Individual coral animals are called polyps, and these polyps reproduce asexually to produce colonial colonies. However, coral reefs also reproduce sexually by spawning gametes, which fertilize in the open water and later develop into larvae. The larvae are free and can swim around before settling on a surface. While the larvae are swimming around, Symbodinaceae colonize in the polyp and form a symbiotic relationship with them before the polyp settles and form into an adult. Once an adult, the coral helps the ecosystem by protecting the coastline from the damaging effects of wave action and tropical storms, provide habitat and shelter for many marine organisms, source of nitrogen and other essential nutrients in the marine food chain, assist in nitrogen and carbon fixation, and help recycle nutrients. This reproductive cycle repeats, often releasing larvae intermittently throughout. When the conditions are right, coral release their endosymbionts, leading to the coral's loss of color. This process is called coral bleaching. Often it is experienced when heat, UV, and/or chemical stressors induce the loss of this photosynthetic algae. Over the years, an increase in temperature has led to global warming, resulting in an increase in the number and intensity of bleaching events experienced worldwide. In the lab, the model organism used to study the conditions of coral bleaching and the genetic basis of tolerance to these environmental bleaching stressors was a species of sea anemone, Exaptasia pallida, which forms a symbiotic relationship with the photosynthetic endosymbiont algae Breviola minutum, unicellular algae in the family Symbodinaceae, allowing the algae to produce nutrients the anemone uses, and in turn, the anemone offers the endosymbiont protection. This particular species of anemone can be bleached and still survive, allowing us to study the genetics of both species a year. In order to have enough species to study the effects of stressors on the organisms, the anemones were cut in half. When the anemones are cut, a ramete or two genetically identical individuals are made. With enough rametes, a genet or family can be produced. Each ramete can then be exposed to differing stressors to compare it to the control. Since the anemones are genetically similar, mutagenesis, or random mutations caused by enzymes, chemicals, and radiation, are responsible for phenotypic mutations observed. Reverse genetics allows us to look for these phenotypic mutations first before discovering the genetic basis of them. The goal of the lab was to analyze coral bleaching processes and to understand the genetic basis of tolerance to bleaching environmental stressors. In the Breviola minutum mutagenesis experiment, the algae cells were being tested under UV light at different time conditions to cause them to mutate, since UV light usually kills these cells. Uracil was added to the algae plates to keep the cells alive, including URA3, which is necessary for uracil production. URA3 converts 5-FOA into 5-FU, which is a toxic compound that kills the cells. Therefore, an absence of the wild-type 5-FOA through mutagenesis will result in the survival of the cells. Based on what we have learned about these organisms, we hypothesize that in the mutagenesis experiment, the Breviola minutum cells that mutate to not have 5-FOA will survive, while those who have both URA3 and 5-FOA will not survive. We also believe that the cells that had the intermediate treatment conditions, 30 seconds to 1 minute, would have the highest chance of survival since they would have sufficient time under the UV light to mutate, but not be over or underexposed. For the anemone stress challenge experiment, we hypothesize that the anemones with the most stressors will be bleached or lighter in color than the control variable due to a loss in symbionts. This would include the anemone in mentholated seawater and the two anemones in 32 degrees Celsius seawater and 34 degrees Celsius seawater, while the control variable will remain the healthiest according to the coral health chart.
Thank you, Lucia, for that great introduction. Now, I, Jonathan Barrios, will go over the methods we used in order to prove our hypothesis. First, I will go over the UV mutagenesis methods. Each member of the group subjected Breviola minutum, strain SSB01, to UV radiation at varying times. The cells were then plated with Diago's IMK medium, which provided uracil to the cells. This was important to add since we were testing against resistance to 5-FOA, which stops the production of uracil in the cells. The plates were then sealed using pearl film in order to prevent contamination inside the plate. Next, I will be going over the methods for the anemone stress challenge, in which our group used genet PK3. To begin the anemone stress challenge, each group was assigned an anemone and a heat or chemical stress. The anemones were then scored using the coral health chart, which can be seen in Figure 6. The color was a measure of the symbiote density within the anemone. They were then left alone for 1 week to observe the results of their conditions and to collect data. After the week was over, the corals were rescored using the coral health chart. In Figure 4, we can see the anemone stress test assembly. So you can see the conditions. Menthol was the chemical conditions, and 32 and 34 degrees Celsius were the heat conditions. 25 degrees Celsius was used as a control. The next step was to extract the DNA. The Zymo DNA kit was used in order to do so. The first step was to degrade the anemone in order to access the DNA, which was done with the lysis solution and bashing bead tube. The genomic lysis was used to help degrade the cells and bind the DNA to the filter columns. The pre-DNA buffer and gDNA wash buffer were used to remove any more impurities that could be left on the DNA. The final step was to add the DNA elution buffer to cause the DNA to release from the column. The different tubes were used to remove different levels of impurities, and the centrifuge was important in order to separate the layers of debris from the DNA. Figure 7 shows the basic methodology we used in order to extract the DNA. After the DNA was extracted, it was important to amplify the DNA in order to do further analysis. Thus, we performed a PCR, a polymerase chain reaction. The markers used in the PCR was the SCAR markers and the 23S marker. SCAR, which stands for Sequence Characterized Amplified Regions, were used specifically for the anemone. These regions have been already sequenced and vary enough to distinguish different populations of anemones. The SCAR markers also contain intersimple sequence repeats, which is a region of DNA containing a variable number of tandem base repeats, which can be used to learn about the different species of anemone being studied in the entire lab. The 23S ribosomal region is tied to the Augean anemone. It is used to identify different species of Auge using a short piece of DNA. Figure 8 shows the tubes and the Simulkit we used for our PCR amplification. After amplifying our DNA, it was important that we analyze it, and we can see in Figure 9 the two methods we use to analyze our data, which is gel electrophoresis and Sanger sequencing. Briefly, I'm just going to go over what we did for gel electrophoresis. So first we made the gel, and then we poured it into the molds. The ladder was then added as a reference point, and the DNA was added next to the ladder. The gel was run under voltage, and the results were viewed under a fluorescent filter box. After viewing the results for the gel electrophoresis, all that was left was to sequence the DNA. To sequence the data collected from the DNA extraction, a program named Finch was used. Finch is a program that sequences DNA using the Sanger sequencing method. Once that was done, the DNA sequence was cleaned up using the Sequence Messenger program to ensure no unnecessary information was on the sequence. The sequences were then compared using the Muscle program to perform a multiple sequence alignment. Figure 10 shows the results collected from the Finch TV software.
This is Jason, and I will be talking about the results. The first experiment conducted was UV mutagenesis of Breviola minutum, and unfortunately, even though plating was successful, the plates were contaminated, and we were unable to see them. If the plates were not contaminated and they were plated properly, we would have seen some growth. The second experiment, the anemone stress challenge, was broken up into 5 days. On the first day, the anemones were put into a condition and scored. On the second day, the anemones' health chart scores were compared. As you can see, for each condition, the sea anemone lost its symbiote, therefore losing their color. The most color loss was observed in the 34 degrees Celsius condition, and the anemone in the 25 degrees Celsius with menthol water died. From these findings, it can be assumed that the genus PK3 is intolerable to any heat and that chemical stressors may completely kill these anemones and their symbiotes. Day 2 consisted of extracting the DNA from the anemones under the differing stress conditions. After using the Zymo DNA extraction kit, the DNA from the lysed cells should have been in the solution at the bottom of the final collection tube, which was used for the next day. The next day, day 3, polymerase chain reaction, or PCR, was conducted. Using the DNA from the DNA extraction, PCR was run to allow segments of DNA being targeted to be amplified. Primers such as 23S, SCAR3, SCAR4, and SCAR5 allowed for the synthesis of these specific areas on the DNA strand. The amplified DNA was later used for gel electrophoresis and bioinformatics. On day 4, once PCR was successfully completed, gel electrophoresis was done on these DNA samples. This allowed DNA to be separated according to molecular weight. From the gel electrophoresis, it shows that 23S and SCAR3 were most similar in molecular weight, while SCAR3 and SCAR4 also shared one single band, signifying they had at least one molecule of similar molecular weight. Finally, 23S, SCAR3, and SCAR5 also shared one band. On the final day, the analysis of bioinformatics was done, and phylogenetic trees were made. From using Finch TV, 23S and SCAR3 were successful in being sequenced, while SCAR4 and SCAR5 had invalid sequences. This is possibly due to an error in reading the DNA extract, or that not enough DNA was extracted from the anemone to have a significant impact on the readings.
Hi, I'm Maria, and I will be doing the discussion portion of this project. To begin with, science is not perfect, and therefore there were multiple sources of error throughout this semester-long research project. The primary source of error had to do with contamination with the UV mutagenesis plates, which did not allow us to see full results. Also, pipetting errors obviously occurred in regards to the SCAR4 and SCAR5 markers during PCR, gel electrophoresis, and sequencing.
The goal of the UV mutagenesis experiment was to perform reverse genetics on Brevolium minutum by exposing it to UV radiation and then testing it for its resistance to 5-FOA. If things would have gone as planned, we would have expected to see URA3 mutants survive. However, contamination prevented us from seeing real results. For further research purposes, we would expect to employ more aseptic techniques in the laboratory. URA3 mutants would have survived because uracil was included in the recovery medium.
Through this semester-long research, much information was obtained regarding Exoptasia pallida and its symbiote. The primary conclusion reached from this project was that the addition of stress on organisms produces change. When Exoptasia pallida was exposed to varying temperatures, it was clear that some changes occurred. At higher temperatures and under chemical stress, the anemone faced several challenges and released its symbiote. The consequences were drastic, as it led to decreased resources for the anemone and even a color change. 4 ramets in different conditions were tested. There was a control variable at 25 degrees Celsius and 3 treatments in ventilated water, 32 degrees Celsius and 34 degrees Celsius. As shown in the previous results slides, all of the anemone ramets face severe change, as there was a decline in health scores. This supports our hypothesis in which we said that the anemones with the most stressors would be bleached or lighter in color, due to a loss in symbionts. Studies previously done on corals point out the following other potential stressors, such as reduced salinity, solar radiation, bacterial and other infections, and varying seawater temperatures. The research obtained from this experiment could be placed on that list, because it is obvious that temperature causes a decrease or almost a decline in the symbiotic relationship between the anemone and the symbiote, which were the model organisms for this coral bleaching. Research done with both Symbiodinium and Exoptasia pallida point out to an actual shocking discovery, in which this mutant coral symbiote alga may have the ability to actually switch off symbiosis. As previously mentioned, research has shown that mutant alga has the ability to shut off symbiosis. If further research is conducted, perhaps one day we could genetically modify symbiote species, which could potentially solve the coral bleaching crisis. Besides this, our semester-long research project has further connections in science. For instance, Ellison et al. Searched for biomarkers for abiotic stress. HSP70 gene in Exoptasia pallida was proven to increase its expression at high salinities, which is a stressor. Therefore, further research can be done on other genes in Exoptasia pallida to be traced as abiotic stressors in order to further solve the coral bleaching crisis. Also, more discovery about the dinoflagellates or symbiont species is essential to further expand our research. Lewis et al. Proved in a study that each species of dinoflagellate is ecologically distinct, exhibiting specific mutualisms with phylogenetically distinct coral hosts. The possibilities are endless, and it would be nice to find a common ancestry between many species of dinoflagellates. Finally, we could potentially expand our research by using another strain of Brevolium, just like Baylis et al. Did in their study. Genetic variation was found between Brevolium antiligorgium and Brevolium minutum species, and among their symbionts, in chlorophyll content, photosynthetic efficiency, and growth rate. This variation is important to further understand the symbiotic relationship and the further implications that stress has. Genetics behind PCR, gel electrophoresis, and sequencing. The results from these processes indicate that the genes that are in connection to SCAR3 are significant to the tolerance of the Symbiodiniaceae, because it was most similar to the 23S sequence, as shown in Figure 13. Both of these sequences actually survived, and SCAR4 and 5 were not able to be identified.